Hey everyone, this is Dr. Marcon, and this is Chapter 8 on genetics. Before we get into genetics, we have to get into the basic building blocks. What are genes? A gene is a sequence of DNA that codes for a protein. We've heard of chromosomes before, so chromosomes actually are made up of genes and have many different genes. You have two versions of every gene, one from mom and one from dad. So we know that genes are carried on chromosomes. They are the basic functional units of heredity with the ability to be replicated, expressed, or mutated. Long ago, a few hundred years ago, there were studies done by a monk named Johann Gregor Mendel that deduced that each individual has two discrete copies of characteristics that are passed individually to offspring. We now call those two copies genes, which are carried on chromosomes, one from each parent. The chromosomes that we inherit and the two copies of each gene are located on paired chromosomes. Now, as a diploid organism, we have two genetic copies that may or may not encode the same version of that characteristic. So, for example, one individual may carry a gene that determines blonde hair and a gene that determines black hair. So we have structures called alleles. So alleles are gene variants that arise by mutation and exist at relative locations on homologous chromosomes. So in a homologous pair, each chromosome has the same set of genes and then may have different alleles or different versions of a gene. So again, alleles are gene variants that exist at the same relative locations on homologous chromosomes. We have what's known as a dominant allele and a recessive allele. So a dominant allele is what is expressed, whereas the recessive alleles are the latent kind of sleepy trait not expressed in the presence of a dominant allele. So diploid organisms that are known as homozygous for a gene have two identical alleles, one on each of their homologous chromosomes. So we can see these alleles are similar and identical. So this is what's known as homozygous. So for example, we'll have two dominant alleles on the same location. So this is known as homozygous dominant. Or we might have the recessive alleles on the same locations of our homologous chromosomes. This is known as homozygous recessive. So again, they can be homozygous dominant or homozygous recessive. And when we assign letters to the alleles, homozygous dominant, you'll see capital letters, so two capital letters. So for example, homozygous dominant would be uppercase B, or if it was homozygous recessive, it'd be lowercase b, lowercase b. So the dominant allele is capitalized and the recessive allele is lowercase. Now, when we have heterozygous alleles, this means the genotype or the genetic makeup has different alleles for the gene being examined. So if we have a heterozygous situation, we'll have a big B and a little b. Okay, so that is what is known as heterozygous. So two alleles for a given gene in a diploid organism are expressed and interact to produce physical characteristics. When we talk about genotype is the underlying genetic makeup consisting of both the physically visible and the non-expressed alleles. So again, this is the genetic makeup of an individual. Whereas the phenotype is the visible or measurable features of an individual. These are the traits that are expressed by an organism. So when it comes to genotypes and phenotypes, again, we have dominant and recessive alleles. So the dominant allele will reveal itself in the phenotype, in the outward appearance, whether the organism has one or two copies. And again, the dominant allele is expressed using the capital letter. Whereas the recessive allele reveals itself in its phenotype only if the organism has two copies of that allele. So they have to be homozygous recessive in order for that trait to be expressed. So for example, brown eyes are actually dominant over blue eyes. Okay, so brown eyes are the dominant trait over the recessive blue eyes. So if we look at the genotype and the phenotype of eye color, we can see that if someone has the genotype big B, then they for sure will express brown eyes. However, if the person has the genotype that is heterozygous, with a big B and a little b, still that dominant trait will be expressed and that person will still have blue eyes. If the person is homozygous recessive, meaning they have both the little b, so little b, that person will express blue eyes. So as we can see, we describe this as a person being homozygous dominant for brown eyes. The person that is heterozygous is what's known as a carrier. So they carry that recessive alleles, but they still express the dominant allele, which is brown eyes. And we can see that the person who is homozygous recessive will express blue eyes. Okay. So we talked about a carrier. A carrier is an individual who is heterozygous for a particular gene of interest, and they can actually pass on that recessive allele without showing any of its effects. So the dominant alleles can mask the presence of a recessive allele and their phenotype is stronger. So we can't really say just because you have brown eyes, that means you're homozygous dominant. You could also be a carrier, but still express that dominant allele for brown eyes. So unless we do genetic testing, we can't really with 100% confidence say that that person is homozygous dominant versus heterozygous for that dominant allele. So here are some examples of human genetic traits. One is someone being a tongue roller, so whether or not they can roll their tongue. So oddly enough, there are some people that actually can't do it. Here we see Daniel Radcliffe of Harry Potter fame, who apparently can roll his tongue 3 times. So the dominant allele is capital letter R, that meaning that that person is a tongue roller. And the recessive allele is lowercase r, meaning this person will be unable to roll their tongue. So if they are homozygous recessive, the person will not have the ability to roll their tongue. Another trait would be a widow's peak. So a widow's peak is actually that little pointy portion at the hairline level and the forehead. So the dominant allele is a capital letter W, meaning that person will express the widow's peak. And the recessive allele will be the lowercase w, meaning that that person has a lack of a widow's peak. So as you can see, without the widow's peak, the hairline is relatively straight. Another trait is whether a person has a free earlobe or an attached earlobe. We know Brad Pitt and Angelina Jolie used to be married, and they actually had a child. So the dominant trait would be with a uppercase or capital E, meaning that person has a free earlobe. And then the recessive allele would be a lowercase e, meaning that person has an attached earlobe. So we can see Angelina Jolie has that recessive, is homozygous recessive for an attached earlobe. Whereas Brad Pitt has a free earlobe, and we can see that that dominant trait was passed on to their child. I believe that is Shiloh. Another trait would be a hitchhiker's thumb. So if you take a look at your thumb, if you have a straight thumb that sticks up straight, the dominant allele would be a capital letter H followed by a lowercase I. And then the hitchhiker's thumb, where if you point your thumb up, it looks like you're hitchhiking. So that would be, of course, the lowercase hi. Then another trait would be a bent little finger. So a bent little finger, I guess here, would show the capital letter B, followed by a lowercase f, meaning that person has the dominant allele for bent little finger. And then we have the lowercase letters BF for the straight little finger. So I guess a bent little finger is a dominant trait. And then mid-digit hair. Ew. So apparently this is a dominant trait. And if you have the dominant trait expressed by a capital letter M, you have mid-digital hair. Whereas if you have the lowercase m allele, or two of them, homozygous recessive, it would be the absence of mid-digital hair. Although I think it would be different for Asians, considering we don't really have a lot of hair, or the hair is very fine. My favorite, dimples. So if you have the dominant allele, it would be expressed with a capital letter D, meaning you have dimples. And if you don't have dimples, the absence of dimples, you have a lowercase d, or you would be homozygous with two lowercase d's. So just some fun traits that we can see that are being passed on. So what is genetics? Genetics is the study of heredity, and heredity is the passing of traits via genes from parents to offspring. And we can actually use the letters of the alleles to kind of figure out the chances of inheriting a trait. So we can see that we have a pair of alleles coming from parent 1, and then a pair of alleles coming from parent 2. And then from there, we can predict the percentage, or the chances of having an offspring with specific groups or combinations of alleles. So this is what's known as the punnett square. The punnett square is a simple chart method where each square represents a possible allele combination. So, a practice question, we have a gene that controls muscle formation in the tongue that allow people with those muscles to roll their tongues. People who lack those muscles cannot tongue roll. So that would be homozygous recessive. So the ability to roll one's tongue is dominant over non-tongue rolling. So suppose a woman who is homozygous as a tongue roller marries a man who is heterozygous. So by heterozygous, this person is a carrier for tongue rolling. So we're going to say that capital letter R represents tongue rolling, whereas lowercase r represents a non-tongue roller. So what is the predicted phenotypic ratio of tongue rolling among their children? So what you would do is create a punnett square to kind of figure this out, to see, what percentage, or what is the percent chance of having children that are tongue rollers versus children that are non-tongue rollers. So let's try to do this practice problem. So we're saying that mom is homozygous dominant for a tongue roller, and we know that the letter representing tongue rolling is R. So what we're going to do is make a punnett square. So let's do the annotation right here. Draw. Okay. So we're going to say mom is, we're going to first make a square. Okay, with 4 squares. I like to put mom on top, so this is mom up here, and we're saying that she's homozygous dominant for tongue rolling. So the letter representing tongue rolling is R, because she's dominant, and she's homozygous, so she has 2 R's. We're saying the baby daddy is heterozygous dominant for tongue rolling, so he's going to be represented by a big R and a little r. So now we can do our punnett square to see the chances of them having children who either have the trait or do not. So the first square we can see is going to combine the 2 r's. So we have a homozygous dominant square there and another one here. And then we are going to put the big R together with the little r. And again, the big R together with the little r. Okay, so now we can see our Punnett square filled out. Now, out of the 4 squares, 2 are going to be homozygous dominant for tongue rolling. So RR equals 2 out of 4. So that's a 50% chance. So these 2 have a 50% chance of having a child who is homozygous dominant for tongue rolling. And then we can see the other 2 squares. We have big R, little r. So 2 out of the 4 squares have the genotype big R, little r, heterozygous dominant for tongue rolling. So again, we can see that big R, little r equals 2 out of 4. So that means there is also a 50% chance of their children being tongue rollers, but heterozygous dominant. But as you can see, if we just ask the simple question, out of this Punnett square, what percentage of their children are going to be tongue rollers, we can say 100% for that phenotype. But if we're talking about genotypes, being either homozygous dominant for tongue rolling versus heterozygous dominant for tongue rolling, meaning that they're carriers of that recessive gene as well, we can say that 50% would be homozygous dominant, and then the other 50%, they have a 50% chance of their children being heterozygous dominant for tongue rolling. Okay, so that explains how we get the percentages using a Punnett square and knowing the genotypes of the 2 parents. We know that genetics, it is complicated. There are instances when a gene can have more than 2 alleles, when alleles are not completely dominant or recessive, or maybe even when a gene produces multiple phenotypes. And we'll go over some of these complicated cases in the next few slides. So yes, genetics is complicated. So let's explore some more complicated inheritance patterns. We're going to talk about incomplete dominance and codominance. So these are degrees of dominance. Now, with complete dominance, these are when the phenotypes of the heterozygote and dominant homozygote are identical. So due to one allele being completely dominant. So this is complete dominance. For example, widow's peak. We talked about how a widow's peak is showing that distinctive kind of pointy feature at the hairline, whereas a straight hairline means the absence of a widow's peak. We know that is contained on a single gene. However, there are different versions of the genes. We know that big W is the allele, or capital W is the allele for widow's peak, whereas the small lowercase w means you have a straight hairline. So in your genes, you have homozygous dominant with the 2 letter W's, 2 capital letter W's for widow's peak, and also you can be heterozygous for widow's peak. And if you are recessive with the 2 lowercase w's, 2 alleles, that is homozygous recessive, that means you have a straight hairline. So now we'll explore some extensions of Mendelian genetics. We have incomplete dominance. So incomplete dominance is when the phenotype of heterozygotes is an intermediate between the homozygotes. So due to both alleles not being completely dominant. And here the most well-studied example of incomplete dominance in humans occurs in the genes for curly hair. Inheriting a gene for curly hair from one parent and a gene for straight hair from the other parent may give or will give a hair texture that is a blend of the two, which is wavy hair. So as you can see, both alleles are not completely dominant. And then we have codominance. Codominance is when heterozygotes express phenotypes of both alleles due to both alleles being fully expressed. So this is codominance. So we're going to get into more Mendelian genetics. Codominance and multiple alleles, we can see examples of this, especially in our ABO blood types. So we talked a little about blood types in lab. We know that there are genes that express possible alleles for blood typing or possible antigens on the surface of the red blood cells that determine our blood type. So again, blood type is based on a gene with 3 possible alleles. We have gene A, which is dominant, producing type A blood, which we have gene B, which is dominant, producing type B blood, and we know that gene O, or type O blood, is recessive, meaning it has neither antigen A or antigen B on the surface of the red blood cell. So we normally produce antibodies against antigens that we do not have. So for example, someone with type A blood would produce antibodies to which antigen? We know that it would produce antibodies to the B antigen. So if we have type A blood, we have anti-B or antibodies in our blood, meaning we cannot, if a person with type A blood is transfused with type B blood, there would be a reaction. So people with type A blood have only the A antigen. Individuals with only type B blood have the B antigen on the surface of their red blood cells. And individuals that have neither A antigen or B antigen on the surface of their red blood cells are type O. And then we have individuals that have both A and B antigens on the surface of their red blood cells. And we know that these individuals have type AB blood. So A and B alleles are both expressed equally, an example of codominance. Another antigen that we talked about is the Rh factor. So this is another group of antigens on the surface of red blood cells. We were able to research this antigen through studies with using rhesus monkeys. So that is why it's called the Rh antigen. So if an individual has the Rhd antigen on the surface of their red blood cells, they are known as Rh positive. If there are no Rhd antigens on the surface of the red blood cell, then they are known as Rh negative or negative. So let's practice blood type genetics. 3 children recently born in a hospital were accidentally mixed up. I smell a lawsuit. The blood types of the parents involved are given along with the blood types of the infant. Determine which baby belongs with which parents and explain your reasoning for the decisions you made. Okay, so we can use our punnett square to help determine the likelihood of the blood types of the children between 2 parents if we know the genotype of each parent. Before we get into this problem, let's review the phenotypes and the genotypes for blood typing. So first we're going to start off with the phenotypes for the different blood types. So phenotypes would be the outward expression, the different blood types. So the different blood types that we have type A. We have type B. We have type AB. And we have type O. The genotypes, or the genetic makeup of these blood types, and this is, of course, according to your book. For type A, we can use capital letter A, and we can have, capital letter I with the superscript A, and we have two, so we're going to do homozygous dominant IA, or type A can be a carrier of the recessive type O allele, and that would be IA and then little I. Okay, so those are the two possible genotypes for type A blood. Same with type B blood. You can have IB, which is homozygous dominant, or type B can also be a carrier of the recessive allele. So IB, then little I. Now type AB will show codominance of the two alleles, so it would be IA and IB. Okay, so those are the two alleles that make up the genotype for type AB blood. Now type O is homozygous recessive, and that is represented by the two lowercase letters I. Okay, so those are the phenotypes and genotypes for your blood types, and you can find this in your book. So now that we have this knowledge, we can use this to make our punnett squares to see the different scenarios or the different possible combinations of blood types and the children that the parents will produce. So let me erase this. And let's start off with parents number 1. So we can see that mom is type A and dad is type B. Okay, and we already went over the different genotypes for type A and type B blood. So let's first start with the scenario where both parents are homozygous dominant for their different blood types. Okay, so let me draw my punnett square. We'll put mom on top here and dad on the side here. Okay, so mom is type A, so we're going to start off with homozygous dominant. She's going to have IA. And we're going to put dad as homozygous dominant as well for type B. So his genotype is IB. Okay, and then we start doing our combinations. So IA, IB here, IA, IB here. I don't want to zoom. Zoom out. There we go. Sorry guys, that's, I don't want to zoom, I want to draw. Zoom out. Let's see if we can start drawing again. There you go. And then again in this square. Okay, so we do the different combinations, and we can see that the genotype IAIB is in 4 out of the 4 squares. So 4 out of 4, that makes it 100%. Whoops. 100% in this combination that their children will be type AB blood. Or 100% type AB. Okay, so that's something to consider. So definitely the first set of parents, they have the possibility of having type AB blood. Now, if we consider the other combinations for parents number one, let's say, for example, mom, she's type A. She's heterozygous for type A. So her genes, her genotype would be IA and then little I. Whereas dad, he's still homozygous dominant for type B. Oops, sorry, little I. Oop. To redraw that. And that's mom. And then we have dad, who's homozygous for type B. So then we do our Punnett square. So we combine IA with IB. Then here we have IB with little I, that's a carrier. And here we have IA with IB. And again, we have IB here with little I. Now here we can see 2 out of the 4 squares has the genotype for type AB blood. So that's a 50% chance that their child might be type AB blood. And here we can see 2 out of the 4 squares have the genotype for the heterozygous carrier of type B blood. So there are 2 possible phenotypes for this combination. We have type AB blood and type B blood. Okay, so that's a possible that's a possibility with this combination. However, we can see among the babies there is no type B blood, so we can leave that out. So right now, parents parent number 1 or parents set number 1 looks strong to be the possibility of being the parents of type AB blood, but we still have to consider all other combinations. So let's see what happens when we have mom is still a carrier for type A, and then dad is also a heterozygous carrier for type B. So let's erase this. So we have mom still being that heterozygous carrier for type A. So she's IA, little I. This is mom. Mind you, we're still in the first set of parents. And then dad is also heterozygous for type B. Okay, and let's take a look at their combinations. So we have IA, IB in this square, and then we have IB, little I in this square, and here we have IA and little I here, and then here we have 2 little I's. So with this combination, we can see that one square has the genotype IAIB, which is the phenotype for type AB blood. 1 out of 4. So 25% chance of their child being type AB blood. And then in another square here, we have IB little I, which is the genotype for type B blood, so 1 out of 4, has a 25% chance of being type B blood. And here in this square, we have a 1 out of 4 chance being type A blood. And then in this last square, we have homozygous recessive for that type O blood type. Okay, so there's 25% chance. So parent set number 1 has the possibility of producing all 4 blood types. So now we have to go through the other sets of parents to kind of narrow down which child belongs to which parent. Okay, so keep in mind that parent set number 1 has the possibility of producing all 4 blood types. So let's erase. Let's go to parent set number 2. Okay, so parent set number 2, we have mom who is type O, and therefore her genotype is little I, little I. So let's make our Punnett square again. So this is parent set number 2. So mom is homozygous recessive, so she has little I, little I. Dad is type AB blood. So his genotype, he has the IA and the IB allele. And this is basically the only combination that we can make with these two parents. So their offspring will produce, in this square, IA little I, and again, IA little I. And in this square, down on the bottom, IB little I, and again, IB little I. So now we have 2 out of the 4 squares being heterozygous for type A blood. So they have a 50% chance of having a child with type A blood. And then a 50% chance of being heterozygous for type B blood, since 2 out of the 4 squares have that genotype. So again, 50%. Okay, so keep in mind, parents number 2 can only have children that are either type A blood or type B blood. Okay, so we kind of have narrowed it down so far. So parent set number 2, definitely a strong case for having the child with type A. Okay, because this is the only combination that they can use to figure out the blood type of their children, since these are the only genotypes that can combine. All right, so now let's go to parent set number 3. So mom has type B blood. Mom can either be homozygous dominant for type B or she can be heterozygous. So let's start with homozygous for type B. So here's mom here. So her genotype would be IB. And dad, well, he's type O blood, so his genotype can only be little I for type O. Okay, so with this combination, we can quickly see that 100% of this combination will produce children with, will produce children with type B blood. So 100% of this combination, their children will be 100% carriers or heterozygous for type B blood, because that's 4 out of the 4 squares. 100%. Okay, so, but let's see what happens when we have mom, uh, having her genotype be heterozygous for type B. So let's erase this. So we're still with parent set number 3. Let's see when mom is heterozygous for type B. So she would be IB, little I. This is mom. And then dad is still homozygous recessive for type O, which is 2 little i's. Okay, so let's see this combination. We have IB, little I here. And then in this square, we have 2 little i's. And then in this square, we have IB, little I. And then in this last square, 2 little i's. I don't want to zoom in. I want to zoom out. Sorry, that is little I. So now using this combination of genotypes, when mom is heterozygous for type B blood, we can see that 50% of their children have the possibility of being heterozygous for type B. And then 50% chance of their children being heterozygous, being homozygous recessive for type O. My stylus is not working right now. Here we go. So parent set number 3, they make a strong case for being the parents of the child with type O blood, since that is the only possibility with this combination of genotypes. Okay, so to review, parent set number 1, they are more than likely the parents of child Z, because they are the only parents that can come up with type AB blood. So parent set number 1, draw a little line to type AB. Parent set number 2, their combinations, their only 2 combinations can come up with either type A blood or type B blood. So since there are no children with type B blood, we can say that they are the parents of type A, which is child X. And then parent set number 3, with their combinations, they can either have children that are type B blood or type O blood. But since, again, there are no children with type B blood, we can safely say that they are the parents of type O, which is child Y. Okay, so make sure you guys know the different genotypes that make up the phenotypes for blood type. And then using this information, we can make different combinations of the genotypes based on our knowledge of the blood type of the parents to see, what blood type their children can be. Okay? So make sure you are comfortable first with the genotypes of the different blood types and then using the Punnett squares to kind of figure out the percentages of the different blood types or blood type combinations of their children. All right, moving forward. Let's stop annotate. So now that we've discussed codominance and multiple alleles with blood typing, let's get into the next extension of Mendelian genetics. We're going to get into sex-linked traits. We know that determination of sex in humans is dependent on our X and Y chromosomes. If one is male, they have an X and Y chromosome. If one is female, they have two X chromosomes. So that determines your sex. A sex-linked gene is a gene located on either sex chromosome, and usually this occurs on the X chromosome. And we have different patterns of inheritance. The father usually passes X-linked alleles to daughters, and the mother passes X-linked alleles to both sons and daughters. And we have different conditions that express these sex-linked patterns of inheritance. So, for example, we have red-green color blindness. Red-green color blindness, basically, you are not able to see colors, the colors red and green. So on the left, we can see normal tricolor vision, and on the right, we can see a picture of the Earth where red and green are not able to be seen. So this is actually an X-linked recessive disease. So we can actually also practice inheritance patterns with sex-linked genes. Now, can a father who is not colorblind have a son that is colorblind? And what about daughters? Assume the mother is not colorblind but is a carrier. First, we need to determine the genotype of the alleles for each parent. Let's annotate here. A little slow. Okay, so we can draw our Punnett square again. First, let's determine the genotype for mom. So mom, she has an X chromosome and a Y chromosome. Okay, so we know that mom has two X chromosomes. She's a female. So two X chromosomes for mom. Dad has an X and a Y. Okay, now the allele is sex-linked. Mom is a carrier, so that means she is heterozygous. Her alleles are heterozygous. So let's designate the letter B for color blindness. So if mom is heterozygous, meaning she's a carrier, so she will be X uppercase B. Oh, okay, of course. So let's do a clear palette. That's fine. So again, let's draw this out. Mom has two X chromosomes. She's a carrier, so she's going to be big B, little b. So she's a carrier for color blindness. Dad, he is an X and a Y chromosome because he's male. And dad does not have color blindness because we know that color blindness is recessive. So we're going to say he's got a big B on his X chromosome. And here we can determine our inheritance pattern. So the first, we'll have two X's with two big B's. Sorry, guys. You're having some technical issues. Let's try that again. Here we have mom. Here we have dad. Dad is XY. Mom is two X's. Mom is a carrier, so she has a big B. and a little b. Dad is not colorblind, so he will have a big B on his X chromosome. Okay, and the first child will have 2 X's, so it's female, 2 big B's. So not colorblind. Here we have a female, possible daughter. We have a big B and a little b. So this next square would be XY, a son having a big B. And then this square will be another son, XY, and a little b. Okay, so that makes sense. They have a 50% chance of having daughters and a 50% chance of having sons. Okay, let's take a look at the inheritance pattern. We can see that one out of the 2 daughters will be homozygous dominant, not being colorblind. This second daughter has the recessive allele, but she is heterozygous, so she is not colorblind, but she is in fact a carrier. So here we have the possibility of one daughter being a carrier. Okay. Out of the 2 sons, we can see that because mom is a carrier, we can see that out of the 2 sons, one will end up being colorblind. Okay, so here we have one son being colorblind. So yes, to answer that previous question, the father who is not colorblind can have a son that is colorblind, assuming or as only if he marries or has a child with a woman that is a carrier for that sex-linked allele for colorblindness. Okay, and then out of his daughters, he can have a child. Both daughters are not colorblind. However, one will end up being a carrier. So let's erase this. In the next problem, could a couple where the man is colorblind and the mother is a carrier ever have both daughters and sons that are colorblind? What is the chance for both males and females? So let's go back. Let's go to a blank slate. So let's take a look at the genotype of those parents where mom is still a carrier for colorblindness and dad is actually colorblind. So if we draw that out, draw our Punnett square again. So mom is a carrier. So let's put 2 X's because she's female. So she is a carrier. She has a big B and a little b. This is mom. And dad is actually colorblind, so he will have the recessive allele on his X chromosome. So he will be X little b. And then Y because he is male. This is dad right here. So let's take a look at our combinations. So we have our 2 daughters here. In this combination, the first daughter could be a carrier. And in this combination, yes, she could be colorblind. Now, the original question asked, could both daughters be colorblind? We're going to have to say no, because one will be a carrier, whereas the other daughter will have colorblindness. Okay, and then for the sons, let's do our XY chromosomes in this box and an XY chromosome in this box. So here we have a combination of the big B allele there and then the little allele there. So again, the possibility of both sons being colorblind is no, because one will not be colorblind, whereas the other one will be if mom is a carrier. Now, the next question is asking about males and females. So in this combination, we do have 1 out of the 4 boxes. So a 25% chance of a daughter, a female being colorblind here. So this box right here, 25% chance of having a daughter that is colorblind. And then with regards to males, we have 1 box out of the 4. So that is also 25% chance of their son or their male child being colorblind. Okay, so this is how we use our Punnett squares to determine percentages of genotypes and phenotypes. As long as you know the genotypes of the parents, you can write out the Punnett squares to determine patterns of inheritance. So what you need to study first is make sure you know the difference between phenotype and genotype, and then kind of work out the genotypes not only for sex-linked inheritance, but also for other phenotypes that we've talked about, such as the widow's peak, blood types, tongue rolling. So make sure you're comfortable, again, identifying phenotypes versus genotypes, and then using the genotypes to plug into our Punnett squares to work out the percentages for patterns of inheritance. And that is the chapter on genetics.